صباح الخير طلبتنا الأعزاء. Our lecture today will be about the thyroid hormone synthesis, metabolism and regulation. It's very important to understand the thyroid biochemistry as it helps us to interpret the thyroid function test. The thyroid function tests are amongst the most common endocrine requests in clinical practice. Thyroid disease is common, so it is likely to be encountered clinically. The objective of this lecture is to state the biosynthetic pathway that involved in the thyroid hormone synthesis. The second objective is to understand the mechanism that regulate the hypothalamic pituitary thyroid axis and to state the thyroid hormone transport, metabolism and biochemical actions. The thyroid gland responsible for the production of the thyroid hormone, including the T3 and T4, and another set of hormone is the calcitonin. The thyroid hormone are a regulator of the basal metabolic rate, regulator of the growth and the oxygen consumption, the development of the human being. While the calcitonin is secreted from other type of cell, it is not thyroid hormone, but it is synthesized and secreted from the thyroid gland from a specialized cell called the parafollicular or the C cell, and it have a different role. It is a regulator of calcium homeostasis. We have two types of thyroid hormone, the triiodothyronine, which is abbreviated as T3, and the tetraiodothyronine or thyroxine, and it is abbreviated as T4. The chemical structure is 3,5,3 dash or comma, which is the triiodothyronine, the T3, and the 3,5,3 dash 5 dash or comma, that is the tetraiodothyronine. The structure of the thyroxine or thyroid hormone generally is composed from the amino acid tyrosine, and it is coupled to iodine. So it is composed mainly of the amino acid, special amino acid, which is the tyrosine, linked or bound to iodine molecule. What gives the specificity? We call this thyroxine and triiodothyronine or T3 or T4, and we have another one which is inactive. The reverse T3 is the number of iodine that's bound to the tyrosine, and also it depends on the site of bounding. The difference between the T3 and T4 is the T3 is missing the iodine at the 5 position. 5/ position. We see this is the T4. We found that this is the iodine attached at site 3, another iodine bound at site 5. At site 3, comma, we have another iodine, and 5, comma, also we have another iodine attached to it. The T3 اللي نعتبره احنا هو البيولوجيكال اكتيف، راح نشوف انه it miss iodine at this site اللي هو 5 comma. We have another product اللي هو reverse T3 اللي هو biologically inactive. We call it reverse T3 اللي يكون missing of iodine at the position of 5 at this position. إذن generally the structure of thyroid hormone هو عبارة عن tyrosine amino acid bound to iodine. Thyroid hormone synthesis. Site of synthesis usually it occur in the thyroid follicle, which are special cell synthesized epithelial cell of the thyroid hormone arranged in the form of follicle. Inside this follicle we have a lumen. This lumen filled with colloid, and the most important substance that's present in the colloid is a glycoprotein that is called thyroglobin. Which is the site, this thyroglobin is the site of the synthesis and secretion of the thyroid hormone. The first step in synthesis of the thyroid hormone is the iodide trapping. As we know, thyroid hormone are synthesized by the iodination and coupling of two molecule of the amino acid tyrosine. This process depends on adequate supply of iodide. The iodide normally in the diet is absorbed from the small intestine, and then it will reach to the thyroid. The uptake of thyroid to the iodide, it is active uptake by the thyroid gland under the control of the TSH, the thyroid stimulating hormone, by the sodium iodide supporter. So even if the concentration of iodide in the circulation is less than the concentration of the iodide in the thyroid gland, there is a pump or supporter which is called sodium iodide, that it's supporter that concentrate the iodide, trap iodide from the circulation to inside the thyroid gland, where it is stored there, and this process is activated by the thyroid stimulating hormone. Usually the concentration of iodide in the gland is about 20 times its concentration in the plasma, and even sometimes it reaches 100 times or more. The next step is that iodide is converted to iodine. Iodide is converted to iodine within the thyroid gland, and this is controlled by a special enzyme that is called thyroid peroxidase. This is still part of the glycoprotein. This process results in formation of a molecule which is called monoiodotyrosine and deiodotyrosine, mediated also by the enzyme thyroid peroxidase. So iodine bound to the tyrosine either at two sites or at one site. If iodine bound to tyrosine at one site, we call it monoiodotyrosine. If two iodine bound to tyrosine in the thyroglobin, so it is called deiodotyrosine. The bounding between the iodine and the tyrosine within the thyroglobin molecule is mediated by thyroid peroxidase. After that, we have a coupling reaction in which either the molecule that is deiodotyrosine DIT bound to another DIT deiodotyrosine، راح يكون عندنا molecule contain 4 iodine molecule. This is called T4 or thyroxine, or deiodotyrosine bound to the monoiodotyrosine, and the result will be a molecule that contain 3 iodine. This is the T3. Then when the time of release, when the body needs thyroid hormone, then the coupled thyroglobin is phagocytized inside the follicular cell, and by the enzyme protease it will hydrolyze the thyroglobin to release the free thyroid hormone, the monoiodotyrosine, the deiodotyrosine, and the free amino acid. إذا من إحنا البody need thyroid hormone, then there will be phagocytosis of the thyroglobin molecule اللي تحتوي على المونوiodotyrosine, الداي وال T3 and the T4, and in addition to the free amino acid, and there will be release of these content. The T3 and T4 اللي يهمنا هو will enter the bloodstream by passive diffusion, while the monoiodotyrosine will die are metabolized more to release the iodide and the free tyrosine, and they will be reused again. the precursor اللي هو الايودين والتيروسين will be used again, while احنا اللي نحتاجه T3 و T4 will enter the circulation. The transport of the thyroid hormone, we know that it needs a binding protein. About 0.3 only percent of the T3 and 0.03 of the T4 are free. فمعظم الثايرويد هرمون are protein bound. We have three types of the binding protein. We have the main one is the thyroxine binding globulin اللي هو من اسمه في specificity. We have the thyroxine اللي عنده highest affinity for thyroid hormone. And the next one we have the transthyretin or serum albumin, واحيانا كان اسمه سابقا thyroxine binding pre-albumin اللي هو عنده lower affinity but high capacity. Also we have the serum albumin اللي هو the next one is the serum albumin اللي عنده lower affinity for thyroid hormone but it contain large amount يعني reserve مالته يكون عالي. So we have the thyroxine binding globulin we have the thyroxine binding pre-albumin وعندنا الالبومين الهايست affinity هو الثايروكسين binding globulin والأكبر capacity هو الالبومين ويكون عنده عادة الالبومين low affinity. والفري اللي هو biologically active هو 0.3% only of T3 and 0.03 of the T4 فقط اللي هو فري واللي يكون biologically active. If we have a question which one is more available, the thyroid in the thyroxine binding globulin or the thyroid in the serum albumin, طبعا الجواب راح يكون the thyroid in the that is carried by the serum albumin because of its low affinity. How are the thyroid hormone metabolized? The major thyroid hormone are inactivated by removal of iodine by the process of deiodination. The minor pathway is through conjugation. معظم الأشياء we want to get metabolized by is by conjugation in the body. Conjugation by bonding of this molecule with another molecule ممكن تكون glucuronic acid or sulfate, anything that make it more water soluble and facilitate its excretion. So the major pathway is the deiodination by removal of iodine from the molecule so it becomes deactivated, and or the another pathway is by conjugation. The major pathway that use the deiodination is mediated by the enzyme deiodinase. We have three types. We have type 1, type 2 and type 3. Type 1 remove iodine from both of the ring of the tyrosine of the and type 2 and 3 remove iodine only from the outer ring. بالنسبة للtype 2 iodinase it will remove the iodine from the outer ring, بينما type 3 will remove the iodine from the inner ring. Type 1 عنده specificity إنه it require the amino acid selenocysteine. فإذا كان عندنا deficiency in selenium in the body or in the diet, so the thyroid hormone metabolism may be impaired, والسبب هو إنه because the type 1 deiodinase activity will be affected. فإذا عندنا two minerals that are important for the proper thyroid hormone function. The first one is the iodine اللي هو part of the structure, والثاني واحد هو السيلينيوم اللي هو نحتاجه بالtype 1 deiodinase اللي هو important for the metabolism of the thyroid hormone. Whenever we have a diet that is low in iodine or in selenium, we have abnormality in the thyroid function. deiodination اللي important for us اللي هو ال 5 dash or 5 comma deiodination اللي هو removal of iodine من ال outer ring. why it's important؟ لأنه راح ينطينا ال biological active form of of the thyroid hormone يحول لنا T4 to T3. T3 is the more active form of thyroid hormone. وهذا يحتاج type 1 deiodinase. ومثل ما قلنا السيلينيوم is very important for its action. Selenocysteine is part of it. So selenium deficiency of selenium راح يأثر على conversion of T4 to T3. T3 is the most important one. While removal of 5 deiodination of the T4, a loss of the iodine from the inner ring راح ينطينا فد compound اللي هو inactive, we call it reverse T3. So if the iodine is removed from the 5 dash or 5 comma راح ينطينا الactive thyroid hormone اللي هو T3, بينما removal of the 5 from the 5 side 5 of the tyrosine molecule راح ينطينا reverse T3 which is biologically inactive. The control of thyroid hormone. The thyroid stimulating hormone stimulates the synthesis and release of thyroid hormone from the thyroid gland. As we know, secretion is from the anterior pituitary gland and it is controlled by the thyroid releasing hormone, and its level also affected by the level of the circulating thyroid hormone. The effect of thyrotropin releasing hormone is that when its serum T3 or T4 decrease, then thyroid releasing hormone will increase and so stimulate TSH and produce the more thyroid hormone. While when T3 or T4 increase, it will give a negative feedback. Thyroid releasing hormone from the hypothalamus will decrease, and this will lead to decrease in the production of the TSH and T3, T4. This is a schematic representation of the regulation. This is the TRH from the hypothalamus, stimulate the anterior pituitary gland. It will release the TSH from the anterior pituitary gland. TSH stimulate the thyroid, produce T4, T3, and when it increases in the concentration, it will give a negative feedback to the hypothalamus. The main effect of TSH, why it is important, we said that thyroid releasing hormone stimulate TSH, TS will control. What is exactly the role of TSH in the function of the thyroid hormone? It will increase the phagocytosis and processing of iodothyroglobulin، راح ينطينا يحول لنا إياه إلى T3 and T4. ورا ما إنه تكون عندنا T3 and T4 and it is still bound to the thyroglobulin, it will يعني يسوي لنا phagocytosis والprocessing يحول لنا إلى full molecule. Also من البداية هو it increase the uptake and activation of iodide. احنا قلنا انه it enhance the effect of sodium iodide supporter اللي يسوي لنا trapping of iodide to inside the thyroid follicle. Increase the hydrogen peroxide generation اللي هو very important as oxidant. يزيد الثيروغلوبين molecule synthesis and transport، ويزيد الراتيو of T3 to T4 ratio. T3 إلى T4 احنا الأهم عندنا والbiologically active is the T3، فال TSH will increase this value. The thyroid hormone receptor are the main mediator of the thyroid hormone action. They are steroid hormone in nature, and after T3 and T4 enter to the inside the cell by passive diffusion, they will bind to a cytoplasmic binding protein, and then T4 is deiodinated to T3 and it will move to inside the nucleus and bound to the thyroid hormone receptor. The action of thyroid hormone, it affects many metabolic process and increase oxygen consumption. Also, it bound to a specific receptor in the cell nuclei, can change the expression of certain gene. Thyroid hormone is essential for normal growth, normal mental development, for sexual maturation. In addition, it increases the sensitivity of the cardiovascular and central nervous system to a certain mediator, catecholamine, and so it will influence the cardiac output and heart rate. In addition, it affects protein turnover, lipid turnover, and carb metabolism. Thank you.